Hello guys, welcome back to our channel. So we're going to see about some of the important topics of our biochemistry subject. Feel important is de novo synthesis of fatty acids. Here so many number of fatty acids will be there. In that for your syllabus they have given palmitate as a one of an example of a fatty acid that which you are going to synthesize now. See, where exactly the location of your de novo synthesis takes place is it will takes place in the particularly in liver, kidney, adipose tissue, and lactating mammary glands. These are the locations of our de novo synthesis. Okay, so then the enzyme machinery for fatty acid production is located in the cytosomal fraction of the cell. Yes, so whatever the cell cytosomal is there, there the entire enzymes will be available. The main sources are nothing but acetyl CoA. Acetyl CoA is the major source of our de novo synthesis. Okay, and that is a building block of our fatty acid also. So the NADPH provides the reducing equivalence. ATP supplies the energy for fatty acid formation. We know that NADPH and ATP are the important source of our energy. Then majorly what you have to remember is the three steps. These three steps has to be important, which can be carrying more importance. First one is production of acetyl CoA and NADPH, conversion of acetyl CoA to malonyl CoA, reactions of fatty acid synthase complex, FAS complex. This is called as fatty acid synthase complex. The entire reaction will be carried out by this multifunctional enzyme. That is nothing but an enzyme. Okay, so the first step we will be going to see is that the production of acetyl CoA and NADPH. Okay, so the we know that it is a prerequisite for fatty acid synthesis. Where exactly the acetyl CoA will be produced? The acetyl CoA will be produced in the mitochondria. It is produced in the mitochondria of your entire cell. Okay, by how? By the oxidation of pyruvate and fatty acids. It will be produced by the precursor called our pyruvate and other fatty acids. Okay, so because of that, we will be getting some amino acids and ketones. So, however, in mitochondria are not permeable to acetyl CoA. So that see whatever your acetyl CoA will be produced in your mitochondria, the mitochondria will be producing that acetyl CoA, but it cannot send the mitochondria, we cannot send the acetyl CoA out of the mitochondria to entire cell cytosol, because whatever the cytosol is there, there only the fatty acid synthesis will be taking place because all the enzymes are available there. Okay, so that's why whatever the acetyl CoA is there, it is not permeable by the mitochondria. So there is some other another system has been arranged so that the acetyl CoA can easily come out of the mitochondria and reach the cytosol of the cell. How it is means the acetyl CoA will be condensed with oxaloacetate. So in oxaloacetate, it will be condensed to form citrate. It will be converted into citrate within the mitochondria, and that citrate is easily permeable by the mitochondria. So citrate will be nothing but the combination of acetyl CoA and oxaloacetate. Okay, so that easily come out of the mitochondria and reach the cytosol. Once the cytosol will be released, so see in this picture you can easily understand that. So this is nothing but entire is a cytosol. Okay, and this is a mitochondria of your cell where the acetyl CoA is must and should. That acetyl CoA is produced by the pyruvate. In pyruvate and some amino acids are required. Some fatty acids are also required for that. And in presence of pyruvate dehydrogenase enzyme, the pyruvate is converted into acetyl CoA. Anyhow, the single acetyl CoA is not permeable by the mitochondria. So that's why it condenses with the oxaloacetate in presence of pyruvate carboxylate, and in presence of citrate synthase will be converted into your citrate. Now the citrate is easily permeable by the mitochondria. So it will come out of the mitochondria, and once it go out, it will be again break down into our acetyl CoA and oxaloacetate. Along with some energy, ATP is converted into ADP and PI. The acetyl CoA is further used for the synthesis of our fatty acid. And whatever the oxaloacetate is there, it is not something useless. Yes, so that in presence of malate dehydrogenase, it will convert it into malate. See here, it is a first NADPH dependent enzyme reaction. It is a first NADPH dependent reaction where the malate is the major source of our fatty acid synthesis. Okay, so malate will produce so many energy for the synthesis of fatty acid, where again in presence of malic enzyme it will be converted into pyruvate. Okay, and that pyruvate will be again sent back into the mitochondria and the entire process will be taken place. Another source of producing the pyruvate is our glucose. Okay, so we have seen this in our glycolysis where the glucose is converted into pyruvate. That is also a source of our pyruvate where the acetyl CoA can easily produced. This is the first step of your production of NADPH. See, NADPH has been produced. Okay, the production of NADPH and the production of acetyl CoA will be done in first step. So the second step is nothing but formation of malonyl CoA. So whatever malonyl CoA is there, that will be produced by your acetyl CoA itself. The acetyl CoA is carboxylated to malonyl CoA by the enzyme acetyl CoA carboxylate. Okay, so acetyl CoA is there. When you provide some biotic carbon dioxide fixation for the biotic, there the ATP is converted into ADP and PI. Then we will be getting the malonyl CoA. Yes, so whatever the fatty acid synthesis are, for that the major precursor are our acetyl CoA and the second precursor which is important is our malonyl CoA. Okay, so in the third step what happens? The reactions of fatty acid synthase complex. Okay, so total 7 steps are there. So we will be seeing that in detail. Okay, so the first step is nothing but whatever the acetyl CoA has been produced, that acetyl CoA will be going to see here one important enzyme is going to come. So that enzyme is nothing acid synthase enzyme, FAS enzyme. So that FAS is nothing but fatty acid synthase. So this is a complex, it is a multifunctional enzyme where it is capable of doing oxidation, reduction, carboxylation, everything can be capable to done by our FAS. So first one, the molecule of acetyl CoA is there. So this is a structure of your fatty acid synthase, FAS complex. One is cysteine, one part is cysteine, one part is ACP. ACP is nothing but acyl carrier protein. One part and another part. First, what happened in the first step? The acetyl CoA is going to bind on your ACP part, acyl carrier protein part. See, in presence of enzyme called acetyl CoA ACP transacylase, where first one, whatever this SH-CoA is there, that will be released. Okay, so above what happened, the SH will be remained and this entire part is going to come here. Okay, so whatever the CoA is there, that CoA will be released out. CoA will be released out. Now you can see that S, C double bond O and CH3 is attached and the acyl carrier protein part of your fatty acid synthase. Don't get confused. In the first step, what happens? Your acetyl CoA will come and bind to the ACP of your FAS complex. It is a structure of your FAS complex. It will come and bind here. Okay, so where the CoA will be released out, there will be no CoA left. So now whatever the cysteine part is empty now, the ACP is occupied by your acetyl CoA. Happens in the first step only, another subtype is there. This is formed. Then whatever this entire acetyl group is there, now it is not acetyl CoA, it is just an acetyl. Okay, so this acetyl will be transferred from ACP to cysteine. Just transfer. The transfer of acetyl CoA to cysteine part of FAS. See, now this has been converted into this. Now the ACP part is vacant and this is called as acetyl S enzyme. Now, at this stage, it is a second step of your fatty acid synthesis. Now, the malonyl CoA will come in picture. I told you two precursors are required. One is acetyl CoA and malonyl CoA. Acetyl CoA is already available. Now, the malonyl CoA will come into picture. So, from here, whatever the CoA is there, we'll just remove that. Here the malonate, here the malonate of this entirely called malonate. The malonate of malonyl CoA will come and attach to the ACP. Yes, the malonate of malonyl CoA will come and attach to the ACP part of your FAS complex in presence of your enzyme malonyl CoA ACP transacylase. Where see whatever the CoA was there that has been released. Now you can see this entire part. Above your acetyl is there and below your malonate is there. Not malonyl CoA, only malonate. Okay, only malonate because from that we have removed the CoA enzyme. Okay, this is called as acyl malonyl enzyme. Now what happens means, now the entire whatever the acetyl group is there, that group will coming and joining to the ACP part only. Means the entire group of acetyl from the cysteine will come and join to the ACP part. Now the whatever the S is there, only it will remain as HS. Okay, now one more thing is happening here. You can see that the carbon dioxide is releasing. This is carbon dioxide is nothing but from here. Whatever the C double bond was there, that will be released as a carbon dioxide. Now you can see that yes, C double bond O, CH2. This is correct. And the acetyl part is C double bond O and CH3. This is the entire beta keto acyl ACP. See, beta is nothing but this is our functional group. The next group will be called as alpha group. The next carbon will be called as alpha and next to that will be called as beta. Okay, so at beta position we have keto group. That's why it is called as beta keto ACP. Okay, so in fourth step what happens? We know that ketones and aldehydes will undergo reduction to form alcohols. So here at your beta carbon, it will be undergoing the reduction to form alcohols. The ketones will be reduced to alcohols in presence of our NADPH. It is the first year NADPH dependent reaction of your FAS complex. Okay, so this is everything will be same, but instead of ketone, we are getting alcohols, and that can be done by our beta keto acyl ACP reductase enzyme. Now this is your functional group. The next carbon will be alpha. Next to that will be beta carbon. So at beta carbon we have hydroxy. So that's why it is beta hydroxy acyl ACP. So next whatever the step will be there, continuation beta hydroxy acyl ACP. Now it will undergo hydration, dehydration. It will undergo dehydration to release the water molecule. How water molecule is released means here already CH2 is there. Can I write like this? CH and H. We'll write the same thing. Whatever the CH2 is there, we will write it as CH and H. Here already OH is there. You have seen that here already OH is there. Okay, so now they will combine to form water molecule. They will combine to form water molecule and that water molecule will be released out. The water molecule is released out. So when the water molecule is released, there is a imbalance in the bonds of our carbon. So to satisfy that, the double bond will be coming. Okay, so this entire process will be taken place by beta hydroxy acyl ACP dehydrase enzyme. Okay, so now the product will be like enoyl ACP, trans enoyl ACP. So now what happened is the NADPH is converted into NADP. Two hydrogens will be provided here. So two hydrogens will be provided by this, so that one will be satisfied and one will be added here to remove our double bond. Here the hydrogens has been added to remove the double bond. So this is nothing but butyryl ACP. Butyryl means because of four carbon atoms are available. This is called as acyl ACP compound. Now what happened? This all is everything is about your sixth step only. Okay, so now the entire transfer of carbon chain from ACP to cysteine. Now whatever the entire chain is there in ACP, that will be transferred into your cysteine part. Okay, now the ACP part is vacant. Now what happens? Your reaction 2 is nothing but the addition of malonate from malonyl CoA till your sixth step. It will repeat for six times. It will repeat for 6 times, where in each time the two molecules will be added. Okay, so just take a look at here. From your malonyl CoA, 1 and 2 carbons will be added. Whatever the C double bond O is there, that will be anyhow eliminated. In this entire fatty chain, we are getting two carbon atoms from malonate at each step. Okay, so reaction, from one reaction, we are getting two, two carbon atoms. From six times, from one time we are getting two carbon atoms. If we repeat the same thing for 6 times, where two carbon atoms are added, so total 12 carbon atoms will be added. Okay, used. If this entire reaction carried for only one time, two carbon atoms will be added from the malonate. If the same thing repeats for 6 times, then total 12 carbon atoms will be formed in the entire chain. That is only the return. See, whatever C double bond O is there, here that is same. Now, here 13 they have added. Previously it was CH2, CH2, CH3. Now 12 extra they have added because the entire reaction has been taken place for 6 times. Now it is 13, including this also it is 13. Now what happens? In presence of palmitoyl thioesterase enzyme, the water is added, means dehydration will occur. Hydration reaction takes place, where the entire breakage of this enzyme will be taken place, where your entire fatty acid is there, no, that will be separated by your FAS complex. Okay, so entire your fatty acid palmitate is there, that palmitate is separated by the your FAS complex. Now what happens? See, CH3, CH2, this entire C double bond O is there. One more oxygen will be provided by the addition of our water molecule, so that we are getting our palmitate as a fatty acid.